ಪಿ ಯು ಸಿ ಸೆಕೆಂಡ್ ಇಯರ್ ಪಾಸಿಂಗ್ ಪ್ಯಾಕೇಜನ್ನು ಡಿಸ್ಕಸ್ ಮಾಡ್ತಾ ಇದ್ದೀವಿ. ಹೌದಾ, ನಿಮ್ಮ ಜೊತೆ ನಾನು ಮೈಂಡ್ ಮ್ಯಾಪ್ ಅಥವಾ ಏನೋ ಕ್ವಿಕ್ಕಾಗಿ ಚಾಪ್ಟರ್ನ ಗ್ಲಾನ್ಸ್ ಭಾಳ 10 to 15 ಮಿನಿಟ್ಸಲ್ಲಿ ಒಂದು ಇಡೀ ಚಾಪ್ಟರ್ನ ಗ್ಲಾನ್ಸ್ ಮಾಡುವಂಥ ಕೆಲಸನೂ ಏನಪ್ಪ ಅಂದರೆ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಅದ್ರ ಜೊತೆಗೆ ಇಂಪಾರ್ಟೆಂಟ್ ಡೆಫಿನೇಷನ್ಸ್ ಏನಪ್ಪ ಅಂದರೆ ನಿಮ್ಗೆ ಗುರ್ತಿದ್ರೆ ಸ್ಕೋರ್ ಮಾಡೋದು ಹೆಲ್ಪ್ಫುಲ್ ಆಗ್ತದೆ ಅನ್ನೋಂಥ ದೃಷ್ಟಿಯಿಂದ 1 ಮಾರ್ಕ್ ಕ್ವಶನ್ಸ್ ಮತ್ತು 2 ಮಾರ್ಕ್ಸ್ ಕ್ವಶನ್ಸನ್ನು ಡಿಸ್ಕಸ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಹಾಗಾದರೆ ಸೆಕ್ಸುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಇನ್ ಫ್ಲವರಿಂಗ್ ಪ್ಲಾಂಟ್ಸ್ಗೆ ಸಂಬಂಧಪಟ್ಟಂಥ 1 ಮಾರ್ಕ್ಸು 2 ಮಾರ್ಕ್ ಕ್ವಶನ್ಸ್ ನೋಡೋಣ. ಓಕೆ, ವಾಟ್ ಈಸ್ geitonogamy? Okay, pollination between the flowers borne on the same plant is called as a geitonogamy. Okay, what are false fruits? ಸುಳ್ಳು ಹಣ್ಣುಗಳು ಅಂತ ಕರಿತೀವಿ ಅದನ್ನು. ಆ್ಯಂಡ್ ಗಿವ್ ಎಕ್ಸಾಂಪಲ್ಸ್ ಅಂತ ಹೇಳಿದ್ದಾರೆ. ದ ಫ್ರೂಟ್ಸ್ ಡೆವಲಪ್ ಫ್ರಮ್ ದ ಥ್ಯಾಲಮಸ್. ಯಾವ ಹಣ್ಣುಗಳು ಥ್ಯಾಲಮಸ್ಸಿಂದ ಬೆಳವಣಿಗೆ ಹೊಂತಾವು ಅವುಗಳನ್ನು ಫಾಲ್ಸ್ ಫ್ರೂಟ್ಸ್ ಅಥವಾ ಸುಳ್ಳು ಹಣ್ಣುಗಳು ಅಂತ ಕರೀತಾರೆ. ಉದಾಹರಣೆಗೆ ಆ್ಯಪಲ್, ಸ್ಟ್ರಾಬೆರಿ ಮತ್ತು ಕ್ಯಾಶು. ರೈಟ್ ದ ಫಂಕ್ಷನ್ ಆಫ್ ಫಿಲಿಫಾರ್ಮ್ apparatus. Filiform apparatus play an important role in guiding the pollen tubes to penetrate in the synergid. Next, what is emasculation? Emasculation, removal of anthers from the flowers bud before the anther dehisces. Okay, ಇದನ್ನೇನು ಕರಿತೀವಿ ಅಂದರೆ emasculation ಅಂತ ಕರಿತೀವಿ. Papaya plants exhibit zygogamy only. Why? Because papaya plant is a dioecious plant. Male and female flowers are developed in different plants. Male and female flowers are developed in different plants. That's why they are called as what? Xenogamy. What is a ophomixis? Ophomixis is the production of seeds without fertilization. Ophomixis is the production of seeds without fertilization. What is a xenogamy? Pollination between the flowers of two different plants, male and female. They are different. What male and female flowers are present in different plants. That's called as xenogamy. What is double fertilization? Two type of fusion. Okay, xenogamy and triple fusion takes place in embryo sac. The phenomenon is termed as double fertilization. Two types of fusions. One is a xenogamy, and another one triple fusion takes place in an embryo sac. The phenomenon is termed as a double fertilization. What is a megasporogenesis? The process of formation of a megaspores from the megaspore mother cell is called as a megasporogenesis. What is a clastogamous? Flowers are invariably autogamous. Since clastogamous flowers are small, inconspicuous, with their accessory whorls never open, they are invariably autogamous. Invariably they are what? Autogamous. Papaya plants are called dioecious. Why? Dioecious. Why? Because these plants produce male staminate and female pistillate flowers on two different plants bodies. That's why they are called dioecious. What is a free nuclear division of a functional megaspores? The repeated mitotic divisions of a nucleus of the megaspores which is not followed by cytokinesis is called as a free nuclear division of a functional megaspore. Okay, 2 mark questions. Briefly explain the structure of pollen grains. Pollen grain is the male gametophyte. It has pollen wall and pollen cells. Pollen wall is two-layered structure containing a outer exine and inner intine. Exine is hard layer made up of a most resistant organic material called as sporopollenin. Exine has a prominent apertures called germ pores. Intine is an inner wall which is a thin and continuous layer made up of cellulose and pectin. Mature pollen grains contains two cells, vegetative cell and generative cells. Next, vegetative cell is bigger with a large irregularly shaped nucleus. Generative cell is a small with a small nucleus. Differentiate differentiate albuminous and non-albuminous seeds with an example for each. Albuminous seeds. Angiosperm seeds which contains endosperm even at maturity are called albuminous seeds. Angiosperm seed which contains endosperm even at maturity are called albuminous seeds. Then non-albuminous seeds. Angiosperm seeds which do not contain endosperm at maturity are called non-albuminous seeds. Example, maize and wheat for albuminous seeds, and non-albuminous seeds example, bean and groundnut. How does pollination achieved in a Vallisneria and sea grasses? Vallisneria and sea grasses. In Vallisneria, the female flower reaches the surface of water by the long stalk, and the male flower or pollen grains are released on the surface of water. They are carried by water current and reach the stigma and achieve pollination. Then in sea grasses, female flowers remain in submerged in water, and pollen grains are released inside the water. Some of them reach the stigma through water and achieve pollination. Okay, this is a way of fertilization in Vallisneria and sea grasses. ಹೌದಾ? ಏನಪ್ಪ ಅಂದರೆ ಇನ್ ವ್ಯಾಲಿಸ್ನೇರಿಯಾದಲ್ಲಿ ಏನಾಗ್ತದಂದ್ರೆ ಮೇಲ್ ಫ್ಲವರ್ಸ್ ಏನು ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಇರ್ತಾವಲ್ಲ ಅವು ವಾಟರ್ ಸರ್ಫೇಸಲ್ಲಿ ರಿಲೀಸ್ ಆಗ್ತಾವ. ಆಮೇಲೆ ಫೀಮೇಲ್ ಫ್ಲವರ್ಸ್ ಏನಿರ್ತಾವಲ್ಲ ಅದ್ರ ಸ್ಟಾಕು ಕೂಡ ಏನಪ್ಪ ಅಂದರೆ ವಾಟರ್ ಸರ್ಫೇಸಲ್ಲೇ ಎರಡು ಫ್ಯೂಸ್ ಆಗೋದ್ರ ಮೂಲಕ ಪಾಲಿನೇಷನ್ ನಡೀತದೆ. ಇನ್ನು ಸಿ ಗ್ರಾಸಸ್ಸಲ್ಲಿ ಅವು ಮುಳುಗಿರ್ತಾವೆ. ಫ್ಲವರ್ಸ್ ಏನಾಗಿರ್ತಾವೆ ಅಂದರೆ ನೀರಿನ ಒಳಗಡೆ ಮುಳುಗಿರ್ತಾವೆ. ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಆಗಿರ್ಬೋದು ಅಥವಾ ಸ್ಟಿಗ್ಮಾ ಆಗಿರ್ಬೋದು. ತಪ್ಪಿ ಅವು ಏನಾದರೂ ಒಂದಕ್ಕೊಂದು ಸಂಪರ್ಕಕ್ಕೆ ಬಂದರೆ ಪಾಲಿನೇಷನ್ ನಡೀತದೆ. ವೆರಿ ರೇರ್ ಇದು, ತುಂಬ ಡಿಫಿಕಲ್ಟ್ ಏನಪ್ಪ ಅಂದರೆ ಫರ್ಟಿಲೈಸೇಷನ್ ಅಂತ ಹೇಳ್ಬೋದು. What is a pollination? Mention any 4 features of wind pollinated flowers. Transfer of pollen grains to stigma of a pistil is termed as a pollination. This is very important. Definitely. Transfer of pollen grains to the stigma of a pistil is termed as a pollination. ಏನಪ್ಪ ಅಂದರೆ ಪುಂಕೇಸರಗಳು ಶಲಾಕಾಗ್ರವನ್ನು ತಲುಪುವಂಥ ಪ್ರಕ್ರಿಯೆಯನ್ನೇ ಪರಾಗಸ್ಪರ್ಶ ಅಂತ ಹೇಳಿದ್ವಿ. ಪಾಲಿನೇಷನ್, ಪರಾಗಸ್ಪರ್ಶ. ಫೀಚರ್ ಆಫ್ ವಿಂಡ್ ಪಾಲಿನೇಟೆಡ್ ಫ್ಲವರ್ಸ್. ಹೌದಾ, ಗಾಳಿಯಿಂದ ಏನಪ್ಪ ಅಂದರೆ ಪರಾಗಸ್ಪರ್ಶಕ್ಕೆ ಒಳಗಾದಂಥ ಫ್ಲವರ್ಸ್ನ ಏನಪ್ಪ ಅಂದರೆ ಮುಖ್ಯ ಲಕ್ಷಣಗಳನ್ನು ಬರೀರಿ ಅಂತ ಹೇಳಿದ್ದಾರೆ ಇಲ್ಲಿ. ದ ಫ್ಲವರ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಎನಾರ್ಮಸ್ ಅಮೌಂಟ್ ಆಫ್ ಅ ಪೋಲನ್ ವೆನ್ ಕಂಪೇರ್ಡ್ ಟು ದ ನಂಬರ್ ಆಫ್ ಓವಿಲ್ಸ್ ಅವೈಲೇಬಲ್ ಫಾರ್ ಪಾಲಿನೇಷನ್. ದ ಪೋಲನ್ ಗ್ರೈನ್ಸ್ ಆರ್ ಲೈಟ್ ಆ್ಯಂಡ್ ನಾನ್ ಸ್ಟಿಕ್ಕಿ, ಹಗುರವಾಗಿರ್ತಾವೆ. ಆಮೇಲೆ ಏನಪ್ಪ ಅಂದರೆ ನಾನ್ ಸ್ಟಿಕ್ಕಿ ಲೊಳೆಯಾಗಿರೋದಿಲ್ಲ. ದ ಫ್ಲವರ್ಸ್ ಆಫನ್ ಪೊಸೆಸ್ ವೆಲ್ ಎಕ್ಸ್ಪೋಸ್ಡ್ ಸ್ಟೆಮೆನ್ಸ್. ಸ್ಟಿಗ್ಮಾ ಇಸ್ ಲಾರ್ಜ್ ಆ್ಯಂಡ್ feathery. Wind pollinated flowers often have a single ovule in each ovary. Numerous flowers packed into an inflorescence. ಹೌದಾ, ಹಲವಾರು ಹೂಗಳು ಏನಾಗಿರ್ತಾವೆ ಅಂದರೆ ಒಂದು ಗೊನೆಯಲ್ಲಿ ಪೋಣಿಸಲ್ಪಟ್ಟಿರ್ತಾವೆ. ವಾಟ್ ಆರ್ ಪಾರ್ಥೆನೋಕಾರ್ಪಿಕ್ ಫ್ರೂಟ್ಸ್? ಮೆನ್ಷನ್ ಪ್ಲಾಂಟ್ ವಿಚ್ ನ್ಯಾಚುರಲಿ ಪ್ರೊಡ್ಯೂಸ್ ಪಾರ್ಥೆನೋಕಾರ್ಪಿಕ್ ಫ್ರೂಟ್. A seedlet fruits formed by the process of parthenogenesis are called parthenocarpic fruits. Seedless fruits formed by the process of parthenogenesis are called parthenocarpic fruits. Banana, a plant which naturally produces parthenocarpic fruit. Next, ಬೀಜರಹಿತ ಸಸ್ಯಗಳು ಪಾರ್ಥೆನೋಕಾರ್ಪಿಕ್. ಡಿಫ್ರೆನ್ಷಿಯೇಟ್ ಬಿಟ್ವೀನ್ ಹೋಮೋಗೆಮೆಟ್ಸ್ ಅಥವಾ ಐಸೋಗೆಮೆಟ್ಸ್ ಅಂತ ಕರಿತೀವಿ, ಹೆಟ್ರೋಗೆಮೆಟ್ಸ್ ಆ್ಯಂಡ್ ಐಸೋಗೆಮೆಟ್ಸ್. ಹಾಗಾದ್ರೆ ಹೋಮೋಗೆಮೆಟ್ಸ್ ಮತ್ತು ಹೆಟ್ರೋಗೆಮೆಟ್ಸ್ ನಡುವಿನ ಡಿಫ್ರೆನ್ಸ್ ನೋಡಬೇಕು. Gametes where sperm and egg are morphologically similar in species are called isogametes or homogametes. Types of gametes where sperm and egg are morphologically dissimilar in species are called what? Heterogametes. ಅದನ್ನೇನು ಕರಿತೀವಿ? ಅನ್ಐಸೋಗೆಮೆಟ್ಸ್ ಅಥವಾ ಹೆಟ್ರೋಗೆಮೆಟ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಹಾಗಾದರೆ ಹೋಮೋಗೆಮೆಟ್ಸ್ ಎಕ್ಸಾಂಪಲ್ಲು ಕ್ಲಾಡೋಫೋರಾ, ಎನ್ ಅಲ್ಗಾ, ಇಟ್ಸ್ ಎನ್ ಅಲ್ಗೆ. ದೆನ್ ಏನಪ್ಪ ಅಂದರೆ ಹೆಟ್ರೋಗೆಮೆಟಿಕ್ ಎಕ್ಸಾಂಪಲ್ಲು ಫ್ಯೂಕಸ್, ಇಟ್ಸ್ ಆಲ್ಸೋ ಅಲ್ಗಾ, ಆ್ಯಂಡ್ ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್ಸ್ ಆಲ್ಸೋ. ವಾಟ್ ಈಸ್ ಅ polyembryony? Mention 2 examples of plants where polyembryony is observed. The phenomenon of development of more than one embryo in a seed is called as an polyembryony. Okay, mango and citrus are examples for polyembryony. Mention the four stages of embryogeny in a dicotyledonous embryo. Mention the four stages of embryogeny in dicotyledonous embryo. Proembryo, globular embryo, heart-shaped embryo, matured embryo. These are the four types of four stages of embryogeny in a dicotyledonous embryo. Differentiate between perisperm and pericarp. Perisperm and pericarp. Perisperm, a thin rudimentary layer of a persistent nucleus still remaining in seed even after fertilization, is called as a perisperm. Pericarp, the complete wall of the fruit is called a pericarp. Then 3 more questions. Describe the outbreeding devices that prevent the autogamy. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. These devices are what adaptations are called as the breeding devices. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Some plants exhibit a self-incompatibility. It is a genetic mechanism and prevents self-pollen from the same flower or other flowers of the same plant from fertilizing the oil by inhibiting pollen germination or pollen tube growth in the pistil. Some plants produce unisexual sexual flowers. If both male and female flowers are present on the same plant, such as a castor and mace monoecious, it prevents autogamy but no gytonogamy. In several species, such as a papaya, male and female flowers are present on different plants that each plant is either male or female. Okay, diarchy. This condition prevents both autogamy and gytogamy. ಓಕೆ ಇಲ್ಲೇನು ಮಾಡಿದಾರಪ್ಪ ಅಂದರೆ ಪಾಲಿನೇಷನ್ನ ಏನು ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಆಗೋದ್ರಿಂದ ಒಂದು ಸ್ವಲ್ಪ ಏನು ವೆರೈಟೀಸ್ ಅಥವಾ ಏನೋ ಪರಿಸರಕ್ಕೆ ಹೊಂದಿಕೊಳ್ಳುವಂಥ ಗುಣಲಕ್ಷಣಗಳು ಏನು ಅಡಾಪ್ಟೇಬಲ್ ಕಂಡೀಷನ್ಸ್ ಅಂತ ಕರಿತೀವಲ್ಲ ಅವು ಉಂಟಾಗೋದು ಕಡಿಮೆ ಇರ್ತದೆ. ಅದಕ್ಕೆ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಆದರೆ ಉತ್ತಮ ಅನ್ನೋದಂಥ ದೃಷ್ಟಿಯಿಂದ ಸಸ್ಯಗಳು ದೇ ಅಡಾಪ್ಟ್ ಸಮ್ ಡಿವೈಸಸ್, ದೋಸ್ ಆರ್ ಕಾಲ್ಡ್ ಬ್ರೀಡಿಂಗ್ ಡಿವೈಸಸ್. ಹಾಗಾದರೆ ಆ ಬ್ರೀಡಿಂಗ್ ಡಿವೈಸಸ್ಸನ್ನು ಹೆಂಗೆ ಬಳಸ್ಕೊಳ್ತಾವ ಅನ್ನೋದು ಇಲ್ಲಿ ಏನು ಮಾಡಿಕೊಂಡಿದ್ದಾರಪ್ಪ ಅಂದರೆ 4 ಪಾಯಿಂಟ್ಸಲ್ಲಿ ಏನಪ್ಪ ಅಂದರೆ ಒಂದೊಂದು ಸಲ ಸ್ಟೈಲು ಮತ್ತು ಸ್ಟಿಗ್ಮಾ ಒಂದು ಇನ್ನೊಂದರಿಂದ ದೂರ ಇರ್ತಾವೆ. ಇನ್ನೊಂದು ಸಲ ಏನಪ್ಪ ಅಂದರೆ ಆ್ಯಂಥರ್ಸು ಮೊದಲು ಬಿಡುಗಡೆ ಆಗ್ತಾವೆ, ಆಮೇಲೆ ಏನಪ್ಪ ಅಂದರೆ ಸ್ಟಿಗ್ಮಾ. ಹಿಂಗೆ ಏನಪ್ಪ ಅಂದರೆ ಒಂದಕ್ಕೊಂದು ಅದೇ ಫ್ಲವರು ಅಥವಾ ಅದೇ ಪ್ಲಾಂಟ್ನ ಬೇರೊಂದು ಫ್ಲವರ್ಗೆ ಫ್ಯೂಸ್ ಆಗದಿರೋ ಹಾಗೆ ನೋಡ್ಕೊಳ್ಳಿಕ್ಕೆ ಏನೇನು ಮೆಥಡ್ಸನ್ನು ಮಾಡ್ಲಿಕ್ಕೆ ಸಾಧ್ಯ ಐತಿ ಅದನ್ನೆಲ್ಲನೂ ಅವು ಏನು ಮಾಡ್ತಾವೆ ಅಂದರೆ ಮಾಡ್ತಾವೆ. ಅದನ್ನೇ ಇಲ್ಲಿ 4 ಟೈಪ್ಸಲ್ಲಿ ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ದಾರೆ. ವಾಟ್ ಆರ್ ಕ್ಲೈಸ್ಟೋಗ್ಯಾಮಸ್ flowers? Write their significance. Okay, then Cleistogamous flowers which do not open at all leads to compulsory self-pollination. Significance of a cleistogamy. Cleistogamous flowers are invariable autogamous as there is no change of allogamy. Then Cleistogamous flowers produce azure seeds set even in the absence of pollinators. Write the 3 advantages offered by the seeds to angiosperms. Seeds offer several advantages to angiosperms. Angiosperms ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ. Angion refers to capsules, sperma refers to seed. Okay, capsule seeds are called as angiosperms. ಹಾಗಾದರೆ ಅದರಲ್ಲಿ ಏನಪ್ಪ ಅಂದರೆ ಆಂಜಿಯೋಸ್ಪರ್ಮ್ ಸೀಡ್ಸ್ ಏನಿರ್ತಾವಲ್ಲ ಅವು ಹಲವಾರು ಅಡ್ವಾಂಟೇಜಸ್ ಕಾರಣ ಆಗಿದ್ದಾವೆ. ಯಾವ್ಯಾವು? Since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Then next, seed have a better adaptive strategies for dispersal to new habitats and help the species to colonize in other areas. Seeds have a sufficient food reserves. Young seedlings are nourished until they are capable of photosynthesis in their own. Uh, the hard seed coat provides a protection to the young embryo. Being a products of a sexual reproduction, they generate a new genetic combinations leading to the variations. Seeds in the basis of our agricultural. Seed is in the basis of our agriculture. These are the seeds, advantages of seeds of angiosperms. Define autogamy. Write two different kinds of flowers that exhibit autogamy. Transfer of a pollen grains from anther to stigma of the same flower. It is called as autogamy. Two different kinds of flowers that exhibit autogamy are chasmogamous flowers and another one clistogamous flowers. What is artificial hybridization? By which technique it is achieved? Crossing a different species to the combine desirable characters to produce. Crossing a different species to combine a desirable characters to produce commercially superior varieties. Okay, this technique is called as a artificial hybridization. Then how the technique what achieved means emasculation and bagging. Emasculation and bagging. Okay. Then next, what is a ophomixis? Mention its significance. Ophomixis, mention its significance. Okay, they given here a mature embryo sac. Okay, it's a what chalazal end. Then anti what poles on head side. Polar nucleus are there. Central cell is there. Egg is there. Then synergids are there. Filiform apparatus is there. Then microphylar end is there. What is a apomixis? Mention its significance. The process of development of a seed without fertilization is called as a apomixis. The process of development of a seed without fertilization is called as a ophomixis. Significance. Ophomixis is useful in production of hybrid seeds and hence it is significant in hybrid seed industries. Name the plant which produces tallest flowers. Amorphophallus. Amorphophallus. 5 mark questions. Describe the structure of mature embryo sac of angiosperm. Mature embryo sac is the female gametophyte of angiosperm. At chalazal end, it contains 3 antipodal cells. At middle, it contains 2 polar nuclei or secondary nuclei. At microphylar end, it has egg apparatus which includes an egg surrounded by 2 synergid cells. At the base of the synergid cell, finger-like structures called as filiform apparatus helps for pollen tube entry into embryo sac. Thus, the mature embryo sac is 7 called and 8 nucleated structure. 8 nucleated structure. ಆ ಸ್ಟ್ರಕ್ಚರ್ನ ನಾವಿಲ್ಲಿ ಏನು ಮಾಡಿದ್ವಿ ಅಂದರೆ ಸ್ಟಡಿ ಮಾಡಿದ್ವಿ ನೋಡ್ರಿ, ಇದೇ ಸ್ಟ್ರಕ್ಚರ್. ಓಕೆ, ಅಲ್ಲಿ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ನ ಅಭ್ಯಾಸ ಮಾಡಿದ್ವಿ, ಇದೇ ಸ್ಟ್ರಕ್ಚರ್ ಅದು. ನೆಕ್ಸ್ಟ್, write the characteristic features of insect pollinated flowers. Characteristics of insect pollinated flowers. The flowers are large, conspicuous, colored and showy. The flowers are large, conspicuous and colored and showy. Next, when flowers are small, they aggregate in the form of an inflorescence. The flowers have a fragrance and sweet nectar. Pollen grains are rough and surrounded by a sticky substance called pollen kit. Flowers are very good places for the insects to lay their eggs. Write events of development of a female gametophyte embryo sac in correct order. Inside the ovule, initially Microspore mother cell divided by meiosis to form a 4 haploid cells called as megaspores. Out of these megaspores, 3 cells disintegrate. Only one functional megaspore is retained. Nucleus of this functional megaspore undergoes 3 rounds of mitosis to form 8 nuclei. This structure with 8 nucleated condition is called as an embryo sac or female gametophyte. Out of 8 nuclei, 3 goes to chalazal end to form 3 antipodal cells. 2 remain at a center to form secondary nuclei. 3 nuclei goes to micropyle and middle one become egg. On both side they form synergid cells. The embryo sac is 7 celled or 8 nucleated structure. 7 celled and 8 nucleated structure. Explain the transverse section of young anther with a labeled diagram, they asked. Okay, this is the anther. Okay, here are the some parts. In abu, sporogenous cells are there, vascular strand is there, connective tissues are there, then middle layer, this part, middle layer, then tapetum, it's in a round shaped, then edges, okay, epidermis is there, outer layer, then endothelium, then middle layer, microspores, it's a mother cell, then tapetum. Okay, these are the main parts of a transverse section of a young anther. Young anther is generally surrounded by a 4 wall layers, the epidermis, then endothelium, middle layer, and the tapetum. The outer 3 wall layer perform the function of a protection and help in uh, dishyacence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupy the center of the each macrosporangium. The next structure, explain the structure of megasporangium. Megasporangium. Okay, main parts are what? Hilum, funicle, here, hilum, funicle, then next micropyle, then micropylar pole, outer integument, then inner integument, then nucleus, embryo sac, then uh, corazal pole. Next one, which one? Chalazal pole. Sorry, here chalazal pole is there. Okay, we have to see their functions. Funicle. It is the stalk of the ovule. Funicle is the stalk of the ovule attaching to the wall of the ovary. It's attaching to where? Here. Attaching to the wall of the ovary. Then hilum, upper part, whatever is shown there. It is the region. Very sorry. It is the region where body of the ovule fuses with the funicle. Next, last one, chalaza. It is the basal part of the ovule. Opposite end of the chalaza is called as micropylar. Next, integuments. These are the ovule membranes. Later they become a seed coat at a micropylar. Micropylar end they form a hole like structure called as a micropyle, which helps for the entry of the pollen tube. Then nucellus, it is the tissue provides nutrients to the developing embryo sac. Then embryo sac is also called as a female gametophyte. An ovule generally has a single embryo sac formed from megaspore. The next one, describe the structure of dicot embryo with the help of a diagram. So this is a dicot embryo. Okay, here plumule is there, upper part plumule is there. Then these are two, whatever are there, those are what cotyledons. Then hyphocotyl. Okay, this part is called hyphocotyl, this part is called a radicle. Then last part is called as a root cap. Okay, here whatever the part is there, that's called as a root cap. See, this part is a root cap. Then we have to see a typically cotyledonous embryo consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons is the epicotyl, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledons is hypocotyl, that terminates at its lower end in the radical or root tip. The root tip is covered with a root cap. The root tip is covered with a root cap. Next, mention the sorry. Mention what difference between macrosporogenesis and megasporogenesis. Okay, very important. It's a macrosporogenesis and megasporogenesis. It is the process of formation of a haploid macrospores or pollen grains from a pollen mother cell. Then megasporogenesis, it is the process of formation of a haploid megaspores from the from the megaspore mother cells. Then macrosporogenesis, it occurs in a megasporangium or anther. Then megasporogenesis, it occurs in megasporangium or in ovule. During this process, all cells of a sporogenous tissue develops into large number of a pollen mother cells. During this process, only one cell of a nucleus develops into a single megaspore mother cell. The process produces a large number of microspores or pollen grains. The process produces only 4 number of megaspores, out of which 3 will be degenerate. This process leads to the development of male gametophyte. This process leads to the development of a female gametophyte. The process is nourished by a cell of tapetum. This process is nourished by the cell of nucellus. Describe the structure of mature male gametophyte or pollen grain. Okay, mature male gametophyte or pollen grain. These are the important parts of a male gametophyte. See, this is called exine. Then inner part is called intine. Then surface is called germ spore. Then generative cell. This part, whatever is there, generative cell. This is a vegetative cell. These are the main parts of a male gametophyte or pollen grain. The pollen grains are the spherical shaped structures formed in the pollen sac by macrosporogenesis. Pollen grains represent the male gametophyte. The majority of plants pollen grain released at two-celled stage, which are haploid. The larger cell is called vegetative cell and which produce a pollen tube on germination. The smaller cell is generative cell which floats in the cytoplasm of vegetative cell. Generative cell produces two sperm sun germination by mitosis. Vegetative cell is covered by a plasma membrane. Over the plasma membrane there are two layers of a cell walls. The outer cell wall is called exine, which is a thick and made up of highly resistant biochemicals called sporopollenin. Exine gives a protection to the internal parts as it is resistant to heat, acid and alkalis. Exine is followed by an inner thin delicate cell wall called as intine, which is made up of cellulose and pectin. On the surface of exine there are some thin and weak areas called germ spores, which where sporopollenin is not deposited. Through these germ spores, pollen tube form a pollen grain will germinate. Explain the structure of a typical monocot embryo with labeled diagram. With labeled monocot embryo with labeled diagram, they asked. Where they given? Okay, monocot. They're not given. Okay. The monocotyledon embryo consists of a single cotyledon. This cotyledon is in grasses is called as a scutellum, which stores reserve food. Scutellum is situated towards the one side of the embryonal axis. The embryonal axis at its lower end has two root primordial tissues called a radicle, which is going to develop into root system after germination. The radicle along with its root cap is covered by a sheath of tissue called colorza. The portion of embryonal axis above the level of attachment to scutellum is called epicotyle. Epicotyle has a shoot primordial tissue which is going to develop into shoot system after germination. This shoot apex is covered by a leafy sheath called uh, coleoptile. Coleoptile. Then what is autogamy? Explain the devices that the plants have developed to prevent this. The types of pollination where pollen grains are transferred to the anthers to stigma of the same flowers, it is called as autogamy or self-pollination. Following are the devices developed in flowering plants to prevent self-pollination. Unisexuality or dioicy. It is condition where the flowers are unisexual, that is will contain either endoecium or gynoecium, so that there is no scope for self-pollination. Example: castor, maize, papaya. Self-sterility or compatibility. An adaptive condition where if the pollen grain from the anther is deposited on the stigma of the same flower will fail to germinate is called as self-sterility or self-incompatibility. Therefore, in these plants, cross-pollination is the only option. Then, dichogamy. It refers to a condition where the anthers, pollen grains of a androecium and a ori stigma of the gynoecium will mature at a different time and thus avoid self-pollination. There are two types of uh, dichogamy, which include protandry and progyny, a condition where anthers ripen or mature before ori and stigma is called protandry, whereas the condition where ori and stigma ripen or mature before the anther is called protogyny. Then hercogamy. Hercogamy, a condition where the anthers and stigma of the same flower are isolated by some mechanical barrier is called as a hercogamy, a condition where the anthers and stigma of the same flowers are isolated by some mechanical barrier. ಹೌದಾ? ಇಲ್ಲಿ ಏನಾಗ್ತದೆ ಅಂದ್ರಪ್ಪ, ಆಂಥರ್ ಮತ್ತು ಸ್ಟಿಗ್ಮಾ ಇವು ಎರಡು ಬೇರ್ಪಡಿಸಲ್ಪಡ್ತಾವ ಯಾವುದೋ ಒಂದು ಮೆಕ್ಯಾನಿಕಲ್ ಬ್ಯಾರಿಯರನ್ನು ಬಳಸಿ. ನಾನು ರಿಪ್ರೊಡಕ್ಷನ್ ಹೆಲ್ತ್ ಹೇಳ್ಬೇಕಾದ್ರೆ ನಾವು ಬ್ಯಾರಿಯರ್ ಆಗಿನ ಬಳಸ್ತೀವಿ, ಅದ ಬೇರೆ ಬೇರೆ ಇನ್ಸ್ಟ್ರೂಮೆಂಟ್ಸನ್ನು ಬಳಸ್ತೀವಿ ಕೊಂಡಮ್ ಆಗಿರೋದ್ರಿಂದ. ಹಾಗೆ ಇಲ್ಲಿ ಬ್ಯಾರಿಯರನ್ನು ಬಳಸಿ ಏನು ಮಾಡ್ತಾವೆ ಅಂದರೆ ಅವು ಫ್ಯೂಸ್ ಆಗೋದನ್ನು ತಡಿತಾವ. ಅದಕ್ಕೆ ಏನು ಕರೀತಾರಪ್ಪ ಅಂದರೆ ಹೆರ್ಕೋಗ್ಯಾಮಿ ಅಂತ ಕರೀತಾರೆ. ಇನ್ ಸಚ್ ಫ್ಲವರ್ಸ್ ದೇರ್ ಮೇ ಬಿ ಕವರಿಂಗ್ ಆಫ್ ದ ಸ್ಟಿಗ್ಮಾ ಆರ್ ಸ್ಟೆಮೆನ್ಸ್ ಆ್ಯಂಡ್ ಸ್ಟೈಲ್ ಮೇ ಮೂವ್ ಅವೇ ಫ್ರಮ್ ಈಚ್ ಅದರ್ ಟು ಫೇವರ್ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್. ಹೌದಾ? ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ಗಿಂತ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಆಗೋದು ಬೆಟರ್. ಅದನ್ನು ಮಾಡಲಿಕ್ಕೆ ಹೆರ್ಕೋಗ್ಯಾಮಿಯನ್ನು ನಡೆಸ್ತವೆ. ಆಮೇಲೆ ಹೆಟ್ರೋಸ್ಟೈಲಿ. It is the condition where the lengths of the stamen and styles will be extremely different. There may be long styles and short stamens, or short styles and long stamens. Okay, this is called as a heterostyly. Okay, then ಡಯಾಗ್ರಾಮ್ ಇದೆ, ಆದರೆ ಅಷ್ಟೊಂದು ನೀಟಾಗಿಲ್ಲ, ಇರಲಿ. ನೆಕ್ಸ್ಟ್ ಒನ್. Explain the process of double fertilization and triple fusion in angiosperms. When pollen grains germinate on stigma, it produces a pollen tube growing through style, which also carries two sperms formed by the generative cell. When finally pollen tube enters into the embryo sac of ovule, the sperms are released here. During this, lytic enzymes secreted by pollen tube will help to dissolve the integuments and filiform process of synergids will help and guide the what penetration of pollen tube. Penetration of a pollen tube. Okay, here they give one diagram. ನಾವು ಪರಾಗದಲ್ಲಿ ಬೀಜದ ಮೊಳುವಿಕೆಯಲ್ಲಿ ಇದನ್ನು ಅಭ್ಯಾಸ ಮಾಡಿದ್ದೀವಿ 10th ಸ್ಟ್ಯಾಂಡರ್ಡಲ್ಲಿ. ಫ್ಯೂಸಿಂಗ್ ಫಸ್ಟ್ ಮೇಲ್ and female gametes. Here fusing what? Male and female gametes. Then second male gametes. These are what? Second male gametes. Secondary nucleus. Here whatever is there, it's secondary nucleus. It's what? Diploid. Diploid. You know, haploid and diploid. Last one chalansak. It's a coating. Then degeneration. Degeneration of what? Cells. Degeneration of cells. Okay, then endosperm, primary nucleus, it's in what? Triploid form, not haploid, not diploid, it's in triploid form. Okay, then whatever the zygote is there, it is in diploid form. After the entry of sperms into embryo sac, one sperm which is haploid will fuse with the egg which is only haploid to form a diploid cell called zygote 2n, which is the main fertilization or what? Zygamy. The other sperm will move towards the central cell and fuse with the polar nuclei of it to form a triploid cell called primary endosperm nucleus. Primary endosperm nucleus, it is called pen 3n. Triploid cell, it's a. After fertilization, zygote develops into embryo, while a pen primary endosperm nucleus develops a nutritive tissue called endosperm, which takes up the fusion of a nourishing embryo. Since there is a fertilization of two cells by two sperms, that is one egg by one sperm and polar nuclei of a central cell by another sperm, the process is called as a double fertilization. It also called as a triple fusion because pen is formed by the fusion of three haploid nuclei. Pen is formed by, how it's formed? What? Fusion of a three haploid nuclei, that is two polar nuclei and one sperm nucleus. One sperm nucleus. Okay, up to now we discussed about reproduction in flowering plants. Okay, in next clip we are going to discuss about what? Human reproduction. ಮ್ಯಾಕ್ಸಿಮಮ್ ಆಗಿ ಐ ಥಿಂಕ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಇನ್ ಫ್ಲವರಿಂಗ್ ಪ್ಲಾಂಟ್ಸನ್ನು ಕನ್ನಡದಲ್ಲಿ ಹೆಚ್ಚು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಲಿಲ್ಲ, ಇಂಗ್ಲೀಷ್ ಟು ಇಂಗ್ಲೀಷೇ ಹೇಳ್ತಾ ಬಂದೆ. ನೋಟ್ಸ್ ಮಾಡ್ಕೊಳ್ರಿ. ಆಮೇಲೆ ಕನ್ನಡದಲ್ಲಿ ಅದನ್ನು ಆದಷ್ಟು ನೀವೇ ಟ್ರಾನ್ಸ್ಲೇಟ್ ಮಾಡ್ಕೊಂಡು, ಇಲ್ಲದ್ರೆ ನಾನು ಹೆಂಗೆ ಆದರೂ ಇನ್ನೂ ಮೈಂಡ್ ಮ್ಯಾಪ್ ಡಿಸ್ಕಸ್ ಮಾಡೋದೀನಿ. ಆ ಟೈಮಲ್ಲಿ ಸಾಧ್ಯ ಆದಷ್ಟು ನಾನು ಕನ್ನಡ ಹೇಳುವಂಥ ಪ್ರಯತ್ನ ಮಾಡ್ತೀನಿ. ಯಾಕಂದರೆ ಒಂದು ಫ್ಲೋ ಅಲ್ಲಿ ಹೋಯ್ತು ಅಂದರೆ ಒಂದು ಫ್ಲೋ ಅಲ್ಲಿ ಹೋಗುತ್ತೆ ಹಂಗೆ. ಓಕೆ, ಇನ್ನೇನು ಮಾಡಬೇಕಪ್ಪ ಅಂದರೆ ಚಾಪ್ಟರ್ ನಂಬರ್ 3 ಹ್ಯೂಮನ್ ರಿಪ್ರೊಡಕ್ಷನ್ನ ಅಭ್ಯಾಸ ಮಾಡಬೇಕು. ಅದನ್ನು ನೆಕ್ಸ್ಟ್ ಕ್ಲಿಪ್ಪಲ್ಲಿ ಡಿಸ್ಕಸ್ ಮಾಡೋಣ ಅಂತ ಹೇಳ್ತಾ ಥ್ಯಾಂಕ್ ಯು ಸೊ ಮಚ್.